In this video, I'm going to explain the theoretical aspects of sexual reproduction in flowering plants. So let us move to the chapter. The flowering plants are nothing but angiosperms, and in angiosperms, flower is the reproductive organ. The flower is a modified stem meant for the purpose of sexual reproduction in case of angiosperms. Apart from that, the flower is a symbol of many human feelings. It is used for the purpose of ornamentation. It has got aesthetic value. It has a social importance, religious importance. But in the angiosperms, the flower is produced for the purpose of sexual reproduction. The flower consists of 4 parts: calyx, corolla, androecium and gynoecium. Among these 4 floral parts, the calyx and corolla are the non-essential floral whorls or accessory floral whorls. Androecium and gynoecium are called essential floral whorls. Because calyx and corolla are not participating in the reproduction, hence they are non-essential. Androecium is male reproductive part of the flower. Gynoecium is female reproductive part of the flower. So both of them are involved in the reproduction. Hence they are essential floral whorls. And all these floral whorls are located on central axis called thalamus. So typical flower of angiosperm consists of calyx, corolla, androecium and gynoecium. The individual unit of the calyx is sepal. The individual unit of the corolla is petal. Individual unit of androecium is stamen, and individual unit of gynoecium is pistil or carpel. Here, this is the typical stamen. Here you can see the bulged portion, anther. The tip of the stamen consists of anther. Stamen has got a long slender stalk known as filament. What is the condition of this anther in case of angiosperms? The stamen consists the bulged portion called anther. The anther is attached to the long stalk called filament. In angiosperms, the typical anther is bilobed, with each lobe having 2 theca, and it is referred as dithecous. The typical angiosperm anther is bilobed. Each lobe is having 2 theca, and the term used for this explanation is dithecous. Anther is said to be tetragonal, having 4-sided structure. Here in the diagram you can see the 4-sided structure, hence it is referred as tetragonal, and the anther is referred as tetrasporangiate. What are the important terms to remember with respect to typical angiospermic anther? Bilobed, dithecous, tetragonal, and one more term is tetrasporangiate. Why the term tetrasporangiate? Because the 2 microsporangia are located in each lobe of the anther. Therefore, the total number of microsporangia are 4. Hence, the angiospermic anther is called tetrasporangiate. The microsporangia contains diploid mass of cells. They are homogeneous mass of the cells. They are diploid in condition, located in the center of microsporangia. This represents sporogenous tissue, and that sporogenous tissue undergoes microsporogenesis and gives rise to the development of the pollen grain or microspore. That is why the sentence here, the microsporangia develop further and become pollen sacs. Once the microsporogenesis is completed, entire sporogenous tissue is converted into pollen grain. Now the same structure is referred as pollen sac. They extend longitudinally all through the length of an anther and are packed with pollen grains. The microsporangia, it will be converted into pollen sac, and the pollen sacs are containing pollen grains. They are longitudinally extended throughout the length of anther. So this is about the structure of anther. Here in this diagram you can see the transverse section of anther. Here the microsporangia, this is lobe of the anther. In both the lobes you can see the two microsporangia. They are circular in outline. It is connected with the help of connective. Both the lobes are joined with the help of connective. When the single microsporangium is enlarged, it shows the presence of these structures. A typical microsporangium is surrounded by 4 wall layers from outside to inside: epidermis, endothecium, middle layers, tapetum, and in the center of microsporangium, the microspore mother cells are present. And these microspore mother cells are nothing but diploid cells of the sporogenous tissue. Epidermis and endothecium are single layered structures. They are protective in nature and also helps in dehiscence of anther. Below the endothecium, 2 to 3 layers of middle layers are present. They are also protective in nature. Apart from that, they are helping in dehiscence of anther. The innermost layer of the microsporangium is tapetum. The tapetum is a very important layer because it is functioning in different ways. The cells of the tapetum are multinucleate. It contains dense cytoplasm, and very important function of the tapetum is the cells provide nourishment for developing pollen grains. Apart from that, tapetum contains the special cells called Ubisk bodies, and these Ubisk bodies secrete resistant organic chemical compound called sporopollenin, and that sporopollenin forms exine of pollen grain. Along with that, the callase enzyme is secreted by tapetum that separates the cells of pollen tetrad at the time of pollen grain formation. Even tapetum secretes pollen kit, which is oily deposition around the pollen grain normally observed in the pollen grains which are pollinated by the insects. But among these, the one important function that you should remember, it provides nourishment for the developing pollen grains. So this is enlarged view of one microsporangium with all its wall layers. When anther is young, here this is the wrong term, actually it is transverse section of young anther, sorry for that. In the center of microsporangium, there is mass of diploid cells, mass of diploid homogeneous cells. Those cells represents sporogenous tissue. The cell of the sporogenous tissue represents microspore mother cell. One microspore mother cell, which is diploid in condition, it undergoes 2 meiotic divisions, meiotic 1 and meiosis 2. Meiosis 1 produces dyad. The 2 cells are produced, both of them are haploid because meiosis 1 is the reductional division. Meiosis 2, both the cells once again undergo the division and gives rise to the formation of 4 haploid cells. The structure is called pollen tetrad. each cell of the pollen tetrad finally develops into the pollen grain. Therefore, from one microspore mother cell, at the end of microsporogenesis, the 4 haploid pollen grains are produced. This process is called microsporogenesis. And microsporogenesis is initiated by diploid cells called microspore mother cells, and the end products are pollen grains or microspore. Then here it is the diagram of mature anther. The difference you can see in the previous slide, the edges are not ruptured because still the anther is in the young condition. Inside the anther, the microsporogenesis is taking place. Once the microsporogenesis completes, the anther matures and the splitting of the anther occurs with the help of endothecium, epidermis and middle layers. Now here you can see the labeling given is pollen grains. Once the anther matures, it dehisces out and it releases the pollen grains which are haploid in condition and the products of microsporogenesis. Here the detailed information about pollen grain. What is this pollen grain? The pollen grain represents male gametophyte of angiosperms. Remember the term male gametophyte because at the time of pollen germination, the male gametes are produced by this pollen grain. So here it is referred as gametophyte. The pollen grains are generally spherical in structure. The diameter of the pollen grain is from 25 to 50 micron meters, and pollen grains are covered by 2 wall layers. Outer layer is exine, inner layer is intine. Both of them are characteristically completely different. Exine is made up of resistant organic material called sporopollenin, and intine is elastic in nature. It is made up of cellulose and pectin. Why the sporopollenin is resistant organic material? Because it can withstand high temperature, strong acid and even for alkaline exposure. And another very important statement about this one, no enzyme can degrade the sporopollenin is known so far. That means till today there is no example for the enzyme which can break the sporopollenin. That means the sporopollenin is resistant to high temperature, the sporopollenin is resistant to strong acids, even it is resistant to alkali, and enzymes cannot degrade the sporopollenin. That is why the pollen grains are well preserved as fossils. What is the reason behind fossilization of pollen grains? For unlimited period of time, the pollen grains are preserved. They are observed during the excavation. The reason why the pollen grains are fossilized, because the pollen grain exine is made up of the resistant organic material called sporopollenin. But on the exine, some minute apertures or pores are present where the sporopollenin is not deposited, and those pores represent germ pore through which the pollen tube comes out during pollen germination. Then the pollen grain contains cytoplasm, and that cytoplasm is surrounded by thin layer of plasma membrane, and pollen grain contains 2 cells, vegetative cell and generative cell. The vegetative cell is bigger in size, it contains abundant reserve food, and it has got large irregular shaped nucleus. What are the things to remember with respect to vegetative cell? It is bigger cell of the pollen grain, it contains abundant reserve food, and it contains large irregularly shaped nucleus. This is the bigger cell of pollen grain. And the smaller cell of the pollen grain is generative cell, and it is floating in the cytoplasm of vegetative cell. It is spindle shaped and contains dense cytoplasm and single nucleus. The smaller cell is generative cell. It is floating in the cytoplasm of vegetative cell. The shape of the generative cell you need to remember, it is spindle shaped. It contains dense cytoplasm and nucleus. In most of the angiosperms, the pollen grains are released from the anther in the two-celled condition. What is this two-celled condition? The pollen grain containing one vegetative cell and one generative cell, if it is liberated with one vegetative and one generative cell, it is called two-celled stage. And in some cases, the pollen grains are liberated or discharged at three-celled stage. In this case, when pollen grains are liberated in three-celled condition, it contains one vegetative cell, but already generative cell is divided into two male gametes with the mitosis. If the vegetative cell with two haploid male gametes is discharged or released, such pollen grain is said to be three-celled pollen grain. The pollen grains may be liberated at a two-celled stage having one vegetative and one generative cell, or they may be liberated at a three-celled stage having one vegetative cell and two haploid male gametes produced from generative cell. So these are the things with respect to pollen grain. So next, the pollen grains, they have got their own importance. The branch which deals with the study of pollen grains is called palynology. Here question is it necessary to study about the pollen grains? Why such a branch is made for the separate study of pollen grains? Because the pollen grains are producing harmful effects on the human body. Because some pollen grains are producing allergy to the human. Some pollen grains, specifically the pollen grains of Parthenium. The Parthenium is nothing but carrot grass. It causes asthma and other kind of respiratory allergy in the human beings. So it is necessary to know which pollen grain is harmful and which one is harmless. Apart from that, the pollen grains are rich in nutrient, so that the pollen grain are consumed as alternate source of food. As they are rich in the nutrients, they can be used as food supplements either in the form of tablet or in the form of syrup. And it is also claimed that pollen consumption increase the performance of athletes and race horses, and these pollen products are available in the market. In this diagram you can see the pollen product images, especially of bee pollen. That means the pollen grain collected from the flowers which are repeatedly visited by the honey bees. Either it is in the form of syrup or it is in the form of tablet. Why the pollen grains are consumed as the food, alternate source of food? Because they are rich in the nutrients. Hence they can be consumed as the alternate source of food. One more important information with respect to pollen grain. The pollen viability. The pollen viability refers to the ability of pollen grain to perform its function after delivering the male gametes to the embryo sac. What is this pollen viability? It is nothing but the ability of the pollen grain to perform its function. Then what is the function of pollen grain? To deliver the male gametes into the embryo sac. This ability is called pollen viability. And pollen viability, it is varying from one plant species to another plant species, and pollen viability is dependent on two important factors. One is the prevailing temperature and humidity. What is the exact temperature at that period? That temperature decides the pollen viability, and humidity of the atmosphere is the another factor which is also deciding the pollen viability. The two examples I would like to give here. The pollen viability in cereals such as rice, wheat, the pollen viability is only for 30 minutes. Within 30 minutes, the pollen grains lose their viability. Whereas in the members of family Rosaceae, Leguminosae and Solanaceae, the pollen grains remain viable for several months. That means the pollen viability may be for few minutes or it may be for several months after they are released from the anther. These are the things with respect to pollen grain. So here you can see the different diagrams. This is of typical pistil. This is with syncarpous pistil, apocarpous pistil, and this is the diagrammatic representation of anatropous ovule. What is this pistil? It is individual unit of gynoecium, and the gynoecium is the female reproductive part of flower. The gynoecium individual unit is called pistil or carpel, and here the gynoecium may be monocarpellary or it may be multicarpellary. Monocarpellary gynoecium with single pistil, multicarpellary gynoecium with more than one pistil. And two more important terms here. Syncarpous pistils are fused in condition. In the diagram, the example shown is papaver, and when the carpels or the pistils are free in condition, it is apocarpous, and the diagram shown is of Michaelia. The standard or the typical pistil consists of three important parts: stigma, style and ovary. Here stigma, it has got a sticky surface. It serves as a landing platform for pollen grains. That means it receives the pollen grains during pollination. Style is elongated slender part present below the stigma, within which the pollen tube grows. And ovary is basal bulged part of the pistil. Inside the ovary, the small compartments are present called locule or ovarian cavity, which contains the tissue called placenta, from which the ovule or megasporangium arises. So here is the structure of ovule, or it is also known as megasporangium. What are the different parts of ovule? So here in the diagram you can see the all parts of ovule. The stalk of the ovule is called funicle. The junction between funicle and the ovule is called hilum. The ovule is surrounded by two protective coverings or envelopes called integument, outer integument and inner integument. Below the inner integument, the mass of nutritive tissue is present, it is called nucellus, and in the center of the nucellus, the female gametophyte or embryo sac is present. The chalaza represents basal part of the ovule. It is present opposite to the micropyle, and the micropyle is the small opening where both the integuments are not joined with each other, and that micropyle leads to micropylar pole. So all these are the structural details with respect to gynoecium and the megasporangium or ovule. And in the diagram you are observing about anatropous ovule. So here it is the developmental stage of embryo sac. How the female gametophyte or the embryo sac develops inside the ovule. The process of development of functional megaspore is representing megasporogenesis. The process of formation of the functional megaspore is nothing but the process is called megasporogenesis. Where exactly it begins? During megasporogenesis, one diploid cell of the nucellus located near the micropyle, it enlarges its size and it starts functioning as megaspore mother cell. It is the cell of the nucellus located near the micropyle or at the micropylar end. It is diploid in condition. It enlarges its size. It contains the large cytoplasmic content. This cell starts functioning as megaspore mother cell. The megaspore mother cell undergoes first meiosis. It produces two haploid cells. This is nothing but megaspore dyad. The second diagram is representing megaspore dyad. Dyad because two haploid cells are arranged here, and both the cells are repeating second meiosis and gives rise to the formation of group of four haploid cells arranged one above the another. This is called megaspore tetrad. Pollen tetrad, microsporogenesis, megaspore tetrad, megasporogenesis, but here the structure can be also referred as linear tetrad as they are arranged one above the another. So totally 4 cells are produced, but among these 4 megaspore cells of the tetrad, one cell becomes functional and gives rise to the development of the embryo sac, which is called monosporic embryo sac. If 2 cells of the megaspore tetrad gives rise to the development of embryo sac, it is called bisporic embryo sac. If all the 4 cells of the megaspore tetrad undergo the development of embryo sac, it is called the tetrasporic embryo sac. But in our PU syllabus, the mentioned method is monosporic development of embryo sac. In this case, only one cell of the megaspore tetrad, it becomes functional and gives rise to the formation of the female gametophyte or embryo sac. Hence, the development is monosporic. Then what happens? The nucleus of functional megaspore, it undergoes free nuclear divisions. The term is very important. That means only nucleus is undergoing repeated mitotic divisions because it has to keep the same chromosome number, and these divisions are free nuclear divisions. What is the meaning of free nuclear division? It is not followed immediately by cell wall formation. Without cell wall formation, The nucleus of a functional megaspore undergoes mitotic division and finally gives rise to the formation of mature embryo sac. So in the diagram you can clearly see the two nuclei are formed at the opposite pole. Both of them are undergoing second mitosis. 4 nuclei are formed. Still it is in the free nuclear condition. Once again the third mitosis occurs and finally 8 nuclei are formed. Once the 8 nucleated embryo sac appears, now here occur the particular arrangement of cells. The very interesting part about embryo sac in angiosperms is they show the specific arrangement of cells. The particular pattern of cell arrangement, that is characteristic distribution of the cells, can be seen in the embryo sac. What is that? At the chalaza, 3 haploid cells occupy and they represent antipodals. At the micropylar end, again 3 cells are organized or arranged representing egg apparatus. And this egg apparatus consists of 3 structures. All of them are haploid. One is female gamete egg and remaining 2 are synergids. So egg apparatus is located at the micropylar end of embryo sac. The larger cell of the embryo sac is central cell, which is having 2 polar nuclei. They are also haploid in nature. They play important role in the formation of primary endosperm nucleus at the time of double fertilization. One more very important part to remember with respect to synergids is at the base of synergids there is special cellular thickening called filiform apparatus. At the base of the synergid, the cellular thickening is present. It is called the filiform apparatus, and this filiform apparatus plays important role in guiding the pollen tube entry into the synergid at the time of pollination or at the time of fertilization. So these are the structural details with respect to the embryo sac located in the center of the nucellus, also known as female gametophyte, and in angiosperms the mature embryo sac is 8 nucleate and 7 celled structure. 3 nuclei from antipodals plus 2 polar nuclei 5, 1 nucleus of egg and 2 nuclei of synergids. Totally, the mature embryo sac contains 8 nuclei and 7 cells, 3 antipodals, 1 central cell 4, 1 egg cell 5 and 2 synergids, totally 7. So mature embryo sac is 8 nucleate and 7 celled structure. So this is regarding the female reproductive part of the flower, and that is gynoecium. So moving to the next bit, pollination. Transfer of pollen grains to the stigma of a pistil is called pollination. Here, depending on source from where the pollen grains are transferring and which one is the receiving stigma, whether it is belonging to the same flower, whether it is belonging to 2 different flowers produced on the same plant, or belonging to the 2 different flowers produced by 2 different plants. On that condition, the pollination is divided or classified into 3 types: autogamy or self-pollination. Then in the next slide, you can see here geitonogamy and xenogamy. So autogamy, geitonogamy and xenogamy are the 3 types of pollination. In autogamy, the pollen grains from the anther are transferred to the stigma of same flower. So both stigma and the anther are belonging to the same flower. From the same flower, the pollen grains are transferred to the stigma. Such a pollination is called autogamy. And autogamy is not a rule in all the angiospermic flowers. To execute the autogamy, some conditions of the flower are required, such as synchronization in the maturity of the stigma and anther. That means when anther releases the pollen grains, stigma should attain the maturity to receive the transferred pollen grains. The receptivity ability should be there for the stigma to receive the pollen grain and to support the pollen germination. This is nothing but homogamy. The synchronization in the maturity of stigma and anther supports the autogamy. Then, if the stigma and anther are lying close to each other, if they are arranged very close to each other, that also supports autogamy. And another condition, there is no choice at all. Without autogamy, there is no other pollination is possible in the cleistogamous flowers. The cleistogamous flowers are growing on the root. They don't get the sunlight. As a result, they do not open at all. When flower is not at all blooming, there is no question of receiving the pollen grains from other species. Definitely, it is undergoing autogamy without any choice. Apart from that, even though flower is in the closed condition, another condition supporting is the anther and stigma lie close to each other. When the anther dehisces, immediately it gets the stigma, the pollen grains are deposited, autogamy is executed. Therefore, cleistogamous flowers always ensure seed setting or seed formation without the help of any pollinating agent. If the pollinating agent is not available, in some cases the pollination may not take place, but that is not the problem for cleistogamous flowers. As they are in the closed condition, the pollination occurs within the flower and there will be no problem with the seed setting even in the absence of pollinating agents. So this is about the autogamy. So here you can see the two more types of pollination, geitonogamy and xenogamy. Geitonogamy, transfer of pollen grains from anther of one flower to the stigma of another flower. That means here the pollination is occurring between two flowers, but both the flowers are developed on the same plant. Both the flowers are produced on the same plant, but pollen grains from one flower are transferred to stigma of another flower. Therefore, geitonogamy functionally represents cross-pollination, but it is similar to the autogamy as it is not bringing any genetical variations. Genetical variation is not possible because two flowers are involved in the process, agreed, but both the flowers are belonging to the same plant, so that genetical constitution remains same, but geitonogamy requires pollinating agents because pollen grains from one flower are transferring to the stigma of another flower. As a mediator, the pollinating agents are required. Hence, it is cross-pollination. Then in what way it is similar to the autogamy? As it is not bringing any genetical variations, it is similar to the autogamy. Then xenogamy. The genetically different pollen grain and stigma are participating in the xenogamy. Transfer of pollen grains from anther to the stigma of different flowers. Here they are born on two different plants. They are developed on two different plants. Pollen grains of one flower are transferred to the stigma of another flower. Both of them are produced by two different plants. This process is called xenogamy, and here it is 100% dependent on the pollinating agents, because pollen grains must be transferred from one flower to another flower. Somewhere it is developed on another plant, and it is also bringing the genetical modifications. So these are the three types of pollination, and here the xenogamy or cross-pollination, it is dependent on some conditions of the flower. They are all referred as outbreeding devices. The device which discourages self-pollination and which encourage the cross-pollination in a flower, all those devices are called outbreeding devices. And the outbreeding devices observed in the flowers are dicliny or unisexuality. It is the production of unisexual flower, that is monoecious as well as a dioecious condition. For monoecious, the maize is example. For dioecious, papaya is example. This dicliny or unisexuality prevents the self-pollination. Dichogamy, difference in the maturation of anther and stigma. There will be no synchronization in the maturity of stigma and anther. In some flowers, the anthers attain the maturity first. For example, sunflower and rose, it is called protandrous. And in case of the mirabilis and magnolina, the androecium attains the maturity first, which is called protogyny. When the maturity period of stigma and anther differs, this condition is called dichogamy, and dichogamy supports cross-pollination. One more is self-incompatibility. Example is tobacco and mustard. In case of self-incompatibility, even though the pollen grains are transferred to the stigma of same flower, but genetic condition of that stigma is not supporting the germination of the pollen grain of same species. This is nothing but self-incompatibility. One more is heterostyle. It is a condition where the anther and stigma are placed at different levels. Their lengths are not matching with each other. When the length varies, the pollen grain and stigma not come in contact with each other. So autogamy is prevented and the xenogamy is supported, or cross-pollination is supported. So all these conditions or the devices, dicliny, dichogamy, self-incompatibility and heterostyle, all of these are supporting the xenogamy or the cross-pollination. They are called the outbreeding devices. One more device I would like to mention here, hercogamy, a condition where the barriers prevent self-pollination. For example, presence of wings around the pollen grains. They carry the pollen grain away from the vicinity of stigma. In that case, the self-pollination is prevented and cross-pollination is supported. All these devices are termed as the outbreeding devices. So coming to the next part. So here the three images you can observe: wind pollinated plant, water pollinated plant and the insect pollinated plant. Usually cross-pollination is dependent on pollinating agents. It may be biotic pollinating agent or it may be abiotic pollinating agent. The abiotic pollinating agents are wind and water. Biotic pollinating agents are especially animals, and among them the important one is insect. If pollen grains are brought by wind, it is called anemophily, and the flowers are anemophilous flowers. They have got their own characters. They have exposed stamens so that they can release the pollen grains into the wind. The pollen grains are light in weight so that they can be easily carried by the blowing wind. The pollen grains are non-sticky. The stigma is long and feathery so that it can receive the pollen grains which are brought by the blowing wind. And the small flowers are grouped in the form of inflorescence, and one ovary is having only one ovule. These are the some characters that can be observed in case of the anemophilous flower. Then second one, here in this diagram you can see the pollination by water. Pollination by water is called hydrophily, and the flowers are called hydrophilous flowers. And remember, this is restricted or this is observed only in 30% of the genera, and here the male gametes are carried by the water current. What are the characters of water pollinated flowers? The pollen grains are long and ribbon-like. The pollen grains are protected from wetting by mucilaginous covering. The pollen grains in the water pollination are additionally covered by a mucilaginous covering that prevents the pollen grain from wetting. The example, Vallisneria, the same diagram you can see there. It is freshwater plant. Zoostera, it is marine grass. And some plants like lotus and lily or eichhornia, they are also aquatic plants, but for the pollination they are not dependent on the water. In case of Vallisneria, the pollination is taking place on the surface of water. This is called epihydrophily. The term epihydrophily, the pollination is taking place on the surface of water. Here in case of Vallisneria, the female flower is brought to the surface because it is having long stalk, but the male flowers are at the bottom. The male plant is producing the male flowers and it is not at all having the long stalk. These male flowers carrying pollen grains are released in the water. They flow in the water. When they come in contact with the stigma, the speciality of the stigma in Vallisneria is it is trifid. It is having 3 distinct parts. The pollen grain adheres to the stigma and the pollination is executed. Once the pollination is completed, the female flower is brought down into the water and allowed for the fruit setting. This is the specific mechanism. Here you can see the trifid stigma in case of Vallisneria. In case of Zoostera, the condition is opposite. The type of the pollination, water pollination in Zoostera is called hypohydrophily. That is, pollination is taking place below the surface of water. On the surface of water, epihydrophily, it is nothing but in case of Vallisneria. Hypohydrophily in case of the seagrass or Zoostera. Then one more diagram, pollination by insect. When the pollen grains are carried out by insect, it is called entomophily, and the flowers pollinated by the insects are called entomophilous flowers. The entomophilous flowers have Some special characters, very specific. Flowers are large in size. They are very colorful, having bright colors. They are attractive so that they can attract the insect. They have got fragrance. They are rich in nectar. In search of nectar only, the insects visit the flower. Pollen grains are sticky. When insect sits on the flower, the pollen grains adhere to the insect body parts. For that purpose, the entomophilous flowers are always producing the sticky pollen grains. In some species, the floral rewards provide the safe place to lay the eggs. One example given in NCERT, Amorphophallus. It is growing 6 feet in height. Inside that flower, number of insects are laying the eggs. The flower is providing safe place for laying the eggs. In turn, these insects are pollinating. So mutualism, both of them are benefited. Some flowers, to attract the insect, they are producing foul odor. To attract the insect, especially the insects like flies and beetles. Good fragrance as well as foul odor. Both the characters can be observed in the entomophilous flower, which are specially adopted for insect pollination. This is about the anemophily, entomophily and heterophily with specific examples. So then coming to next one, pollen-pistil interaction. This is completely dependent on the chemical components. Once the pollen grains are transferred to the stigma, it doesn't mean that all the pollen grains undergo germination. It is decided by the chemical interaction between stigma and pollen grain. If the pollen grains which are transferred to the stigma are of right type or compatible, the stigma supports the germination of pollen grain. If the transferred pollen grains are of wrong type, that is incompatible, the stigma rejects the pollen grain and those pollen grains are prevented to germinate. All these interactions are together referred as pollen-pistil interaction. Right type of the pollen grain, that is compatible, it is supported by the stigma and allowed to germinate. Wrong type of the pollen grain, that is incompatible, rejected by the stigma, not allowed to germinate. So this is the main theme or process of pollen-pistil interaction. Here in this diagram you can see the pollen tube has entered into the embryo sac through the micropyle. Here it is filiform apparatus. The filiform apparatus guides the entry of pollen tube. Once it enters into the synergid, the pollen tube has released the 2 male gametes. So 2 male gametes are released into the synergid and the filiform apparatus guides the entry of pollen tube. This is possible only after pollen-pistil interaction. Compatible pollen grains are allowed to germinate. The generative cell gives rise to the formation of 2 haploid male gametes. The pollen tube carry those male gametes into the embryo sac to execute a very significant process observed in the angiosperms. It is double fertilization. So the pollen tube, it has carried 2 haploid male gametes into the embryo sac. But the female gamete within the embryo sac is only one. Only one egg cell is present. Then what happens? So let us go for this one. One male gamete move towards the egg cell. The male gamete which is located in the first position at the tip, it fuses with the egg nucleus and result in syngamy or fertilization, gives rise to the formation of diploid structure called zygote. The zygote is the product of fertilization. So one male gamete has completed its function. Then what happens to another male gamete? So one more male gamete is fusing with secondary nucleus. What is that secondary nucleus? Secondary nucleus is a diploid structure produced by fusion between two haploid polar nuclei of the central cell. With that secondary nucleus, another male gamete fuses and gives rise to the formation of that important triploid structure called the PEN, primary endosperm nucleus. As this process involves the fusion of three haploid nuclei, it is triple fusion, and the product of double fertilization and triple fusion. Double fertilization, one is normal fertilization and another is vegetative fertilization. Here the products are, one is a diploid zygote which later develops into embryo, and another is a triploid structure, that is PEN, primary endosperm nucleus. Here one more thing I would like to share. Once the primary endosperm nucleus is formed, now the central cell is referred as primary endosperm cell. Till the polar nuclei are present, it is the central cell. But now it has already completed the double fertilization and triple fusion. Now the central cell containing the primary endosperm nucleus, it is referred as primary endosperm cell. Primary endosperm cell containing primary endosperm nucleus. So here you can see the same explanation. After fertilization, the diagram here you are observing is showing the fertilized embryo sac. The antipodals undergoing degeneration. The synergids are also undergoing degeneration. Now the labeling is not the egg cell. Here the labeling is zygote. The central cell is now referred as primary endosperm cell, and the structure of primary endosperm cell is triploid primary endosperm nucleus. The zygote undergoes division to produce the proembryo that gets converted into globular embryo and heart-shaped embryo, and finally gives rise to the development of mature embryo. So these two diagrams, the dicot embryo and monocot embryo, longitudinal section of monocot embryo. Here the dicot embryo shows two cotyledons, plumule from which the shoot system arises, then radicle from which the root system arises. Radicle is covered and protected by root cap. Other than these structures in the monocot embryo, you can see some additional structures. The scutellum, it is the largest single cotyledon of the monocot embryo. The shoot apex is surrounded by young undifferentiated group of the leaves called coleoptile. Disappearing cotyledon is called epiblast. The radicle is covered by undifferentiated structure called coleorrhiza, both radicle and root cap. These are the specific structures. Scutellum, epiblast and coleorrhiza are observed only in the monocot embryo. So once the fertilization is completed, the ovule gets transformed or converted into seed, and integuments develop into seed coat. Ovary develops into fruit, and wall of the fruit is called pericarp. The seed may contain endosperm. After the complete development of embryo, it is albuminous seed or endospermic seed. If the seed is without endosperm, it is a non-albuminous or non-endospermic seed. Sometimes the seed contains the remnant of nucellus, it is called perisperm in case of black pepper. The fruits may be produced without fertilization. They are called parthenocarpic fruits. In angiosperms, the fruits are the product of fertilization. But sometimes in case of apple and strawberry, the fruits are produced without fertilization. They are called the false fruits, and wall of the fruit is called pericarp. So moving to the last bit of this chapter, apomixis and polyembryony. Some of the special mechanisms that we can observe in case of angiosperms. In angiosperms, without fertilization, the seeds are produced. It is called the apomixis. That is why it is said to be mimicry of sexual reproduction, because without fertilization seeds are not generated. But here the fertilization is absent. That means it is asexual. But still it is producing the seeds which is mimicry of sexual reproduction. It is a form of asexual reproduction because the fertilization is absent. And why it is said to be mimic of sexual reproduction? Because it is producing the seeds. There are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develops into embryo without fertilization. In many citrus and mango varieties, some of the nucellar cells which are diploid surrounding the embryo sac start dividing and protrude into embryo sac and develop into embryos. In such a species, each ovule contain many embryos. They may be developing from the cells of the nucellus. And this condition is called polyembryony. Occurrence of the phenomenon of occurrence of more than one embryo in a seed is referred as polyembryony. Occurrence of more than one seed in the embryo is polyembryony. Formation of seeds without fertilization is apomixis. Development of the fruits without fertilization is parthenocarpic fruits. Like that, one more definition in first chapter, development of new organisms from the female gamete without fertilization is parthenogenesis. And finally, hybrid varieties of several of our food and vegetable crops are being extensively cultivated, and cultivation of the hybrids has tremendously increased the productivity. With the help of this polyembryony and apomixis, the hybrid superior varieties are produced. So this is about the theoretical explanation of sexual reproduction in flowering plants. One thing I would like to share, whichever is the competitive exam, whether you are appearing for Karnataka CET, JEE or NEET. So first go through the detailed theoretical aspect. Don't neglect the NCERT syllabus. You go through the NCERT syllabus, you read the chapters completely, all PCMB. I am not telling only biology, all the PCMB, and make sure that you are very perfect in the theory. Then you can attempt the questions and try to attempt as much as number of questions are possible. Then go for higher order thinking questions, because questions are based on the definitions. Then reasoning question, true or false, match the following, fill in the blanks, diagram based questions, reasoning questions, assertion and reasoning. So for these type of questions, the proper theory knowledge is very important. So go through the NCERT book as well as other NEET preparation books. I am not telling that only NCERT is sufficient, but NCERT, 70% of NCERT answers 70% of the questions can be answered with the help of NCERT theory. So it is a request that you go through NCERT syllabus also. Don't neglect this. I hope it is clear. Thank you very much.